Great, thank you. Well, why don't we make a start then? Firstly, welcome to all of you. Thank you for joining. We're now at 63 participants, so it's fantastic to have such great attendance. It's almost a year to the day since we kicked off the HDR UK's national implementation project on multi-omics and bringing together cohorts from across the UK. And so it's a great time and a great pleasure to have everybody here to reflect and to hear some of the updates on some of the great science that's emerged. Obviously, it would be even greater delight to all be in the same room together to have a big event, but at least it was easy for everybody to get here. You all have a comfy chair and the coffee is hopefully good as well. So some comforts from that. And hopefully this will be the first of several such events, and future we'll be able to do these in person. As we are on Zoom, a few kind of housekeeping things that we should start with. We are planning to record the events and perhaps use some of the recordings for websites and things. So if you don't want to appear on the recording, feel free to turn your camera off. Please could I also ask everyone to be on mute unless they're speaking as well, so that we don't get interruptions from background noise. I can see pretty much everyone's done that already, which is great. You'll have seen at the start of the meeting we had a waiting room system on as well. We will be keeping an eye on that throughout the day, so if you do drop out for some reason or you have to leave and come back, we will do our best to let you in as soon as possible, so there's no delay. You'll see from the agenda we built in some time at the end of each of the short talks we're going to hear for a few minutes of questions. So feel free to type your questions into the chat box. You should be able to see, if you move your mouse to the bottom of the screen, several tabs, several options, one of which is chat. And we'll do our best to answer as many as possible. Ones that don't get answered live, perhaps the presenters may be able to answer after the talk they've given. I should also, as well as welcoming our scientific presenters, which we'll do in a minute, welcome our patient and public representatives. And this is a key aspect of all of HDR UK's work and of the Multi-omics Consortium. And we'll hear later in the day from Amanda and Sarah, who lead our program on PPI&E. And so a warm welcome to those of you who are the lay representatives. And a note to the presenters and people asking questions, therefore, that although this isn't primarily designed as a public engagement event, we'd like the event to be as accessible and open to people who are not living and breathing multi-omics research every day, as many of you are in the scientific community. So we'll do our best to avoid acronyms where possible and to explain things. And we hope you found the lay summaries of the talks that some of the presenters have provided to be a useful way of engaging with the talks. Amy, is there anything else in terms of housekeeping that we need to mention?
I think that's it. Thanks, Adam.
Very good. Okay, well, I will briefly introduce the day then, and you start by perhaps just reminding everybody of what we kind of set out to do when we started this program. The idea really was to capitalize on the incredible strengths we have in the UK in population cohorts and in multi-omics. And by that we mean things that we can measure typically in the blood, like proteins and metabolites or gene levels, for example. And there are many cohorts, and you see them shown here on the map on the left. We now have more than a dozen cohorts from involving participants from Orkney up in the north to Bristol down in the southwest to Norfolk in the east. Several 100,000 participants in these cohorts who all have some genetic data, some sort of multi-omics data, and linkage to electronic health records. This is an incredibly powerful combination, and really the idea of this whole program is to try and conduct analyses of these data, combine them together in various different ways, to harness the power of these different cohorts as well as their diversity. And you see on the right a series of kind of scientific objectives that we have. And hopefully you'll hear from the showcase of talks today. You'll see examples of some of these in action and how we're leveraging some of the cohorts on the left and the data on the left to answer questions here on the right. In terms of the goals of today, as well as the objective, which is really about showcasing some of the exciting science that's going on. And we'll hear talks from people across the country, across different population cohorts and even beyond, who are involved in some of the research that's emerging from this theme. And really, I see 3 C's here today. We'd like to really celebrate our successes, to see some of the great science that's emerging, to challenge ourselves. How can we do better? How can we continue to deliver high quality science, but how can we improve on that? What could we be doing better as a national effort? And thirdly, really to see how we can collaborate more widely. You'll see many examples of science today which has involved multiple UK cohorts, some that involve cohorts from beyond the UK as well. But how can we do better to bring together more data, more cohorts, more investigators? And the set of scientists you'll hear talking today are involved people both involved in the core of the consortium, but also involved in other cohorts, other international efforts as well. So hopefully we can use this day as a springboard to help us improve and collaborate better. Before we get into the science, just to mention there are also other aspects of the consortium as well. You'll hear later, as I mentioned, about our efforts in public and patient engagement and involvement. There's also another core work program, work package of the program, which is about the informatics infrastructure led by Em and Chris, who I think are on the call today. And an important part of this is about understanding what kind of IT requirements, what kind of informatics infrastructure would be optimal for the kinds of research we want to do. To help with that, we're asking the UK multi-omics community to complete a scoping survey. You'll see the link there, and Amy will put it in the chat. And we're going to disseminate this through various means, including emails and the Slack channels, etc. But really what we're aiming to do here is to canvass the community to say, rather than just using existing informatics environments, which may not have been optimally designed for this kind of research, what would you like to see, or how could this, what would the ideal kind of environment look like? So we've designed a relatively short survey. It should take people no longer than 10 or 15 minutes to complete, and just ask for a little bit about your current informatics environment, but also how it could be improved and what you really need if an ideal environment was to be designed. So please do cascade that to your teams and to relevant people. Obviously, the more answers we get, the better we can get a feel for what the community needs. So I'm going to stop there by way of introduction, and I'm going to hand over to the first speaker. Each speaker is going to hopefully be able to share their own slides. So fans of Chris Whitty will be disappointed that I won't be using his catchphrase of "next slide, please" at every couple of minutes. I will hand over, therefore, to the first speaker on the agenda, who is Mike Inouye. Mike is a Director of Research at the University of Cambridge and also Director of the Cambridge Baker Systems Genomics Initiative. And Mike co-leads one of the work packages in the consortium with Chris Yao from Manchester, which is about analysing across multiple domains of multi-omics. So Mike, over to you, please. We do have a reasonably strict timeline today, so I'll warn you when we're running out of time, and make sure we leave enough time for questions. So Mike, over to you.
Okay, great. Thanks, Adam, and thanks everyone for listening to me today. Can I just check the screen? It's on like the full screen mode and not the kind of presenter tools.
That's correct. Yep.
Okay. Great. So I'll be talking today about a project that we've been working on for the past year or so. So genetic imputation of biomolecular traits to enable multi-domain analyses. So I'll try and cast this as sort of broad as I can, given the very diverse mix of people in the audience, patients, public, world-leading scientists. But I'm happy. Anything unclear, just ask me questions at the end. Yeah. Okay, so that's a little bit blurry, unfortunately. So we know that a lot of the diseases out there, right? A portion of them are down to our genes and genetic variation, and that extends beyond just, say, diseases and into even the molecules and molecular levels within us. So proteins that you have in circulation, metabolites, gene transcription, all of these things have some degree of genetic regulation. And at sort of like the simplest sort of model, one of the reasons for that is that the molecular machinery that actually latches down onto the DNA in the nucleus of the cell actually interacts right with the actual base pairs on the DNA. And if those base pairs change, then the efficiency and the kind of way in which those molecular machines latch onto the DNA changes themselves, right? So if you change the kind of internal components of the system, the outputs change, right? So that means that all the downstream biomolecules that come off of the DNA, especially, so RNA, proteins, and other various molecules, they are to some degree regulated by genetic variation. And we want to study that, and we want to study that using big data. So why does that matter? From a much more wide angle perspective, well, understanding the way in which genetics affects molecular levels within us, and to be able to predict how it does that, can explain individual susceptibility to various things, whether they're disease, or explain how individuals manifest different traits, and indeed how different levels of drug targets might vary from individual to individual, and whether that has implications for how one might treat disease, that sort of thing. So understanding the levels of these molecules and the sources of that variation is quite important for all sorts of different reasons. So we want to construct models that use genetics to predict the levels of these proteins, metabolites, etc. So this is not new. A lot of people have done this. I'm not going to go through each one of these, but it's been done for gene expression. It's been done for proteins. It's been done for cellular traits like white blood cell counts, etc. So really, what we're doing here is not like an amazingly novel idea. We're just going to do it, as kind of well and as sort of validated as we possibly can. And importantly, we're going to do it in a single multi-omic cohort, which is the interval study based here in Cambridge. And so the question that we're really asking is, as it's there at the bottom, can we actually construct genetic scores for the metabolomic and proteomic data in particular here, using machine learning? Can we validate those predicted levels of biomolecules externally across cohorts? And can we explore those potential applications? I won't go into the applications in this presentation, but we'll go through those first sort of components there. Okay, and I'll sort of caveat this with, this is not me at the coalface doing this. I am the lowly PI supervising this work. This is all done by the uber talented Yu Xu, a postdoc in the lab who comes from a machine learning and AI background. Indeed, he used to scrape social media profiles at Trinity College Dublin as part of his PhD, and doing sort of personality inference thereof. We sort of brought him over to the dark side to work on health and biomedical applications. So this is his work. So like I said, this is based on the interval cohort. We use data from all sorts of other cohorts as well. NPHS and ORCADES, Fenlin, and we're looking at various sort of platforms. So Olink, which is a proteomics platform, SomaScan, which is another proteomics platform, Metabolon, which is a metabolomics platform, so looking at metabolites, and so on and so forth. And there's a few others that are listed here that we're still in development as well. And we need, importantly here, a training cohort. That's the data in a data-driven way that we construct the models. And then we need a totally independent, external, set of samples, either from the same cohort or from a totally separate cohort, where we then independently test the accuracy, the validity of those of the models that we construct in the training set, right? So that's how all of these sort of data fit together. So I won't get into too much sort of detail here about variable selection, particularly given the broad sort of audience, but we can't sort of look at everything simultaneously. So we have to choose subsets of the actual genetic data of the SNPs that we're looking at. So these are single nucleotide polymorphisms. There are various ways in which we do that. So you can consider, everything. You can consider just those genetic variants that are close to the gene, or enzymes that might regulate the levels of the metabolite that you're interested in, and or you can look at all sorts of different combinations thereof. So we looked at a bunch of different ways in which we consider genetic variants, and we found that looking at the local sort of region around the gene of interest, sort of downstream 1 megabase, upstream 1 megabase, and then considering all the kind of genetic variants that are associated strongly with that particular gene output, genome wide, doing an initial pass, taking all those and then fitting a model on top of that, that was the best approach. So, and that's for the statistical geneticists in the audience, you can see sort of the p-value thresholds at genome-wide significance there as well. So to do the model construction, we used a particularly efficient approach that we found, because we were looking at thousands of traits. We can't necessarily use like really deep neural nets or anything like that, where you have to do a whole lot of hyperparameter tuning. We chose Bayesian Ridge, where you can utilize, it's fairly straightforward to do hyperparameter tuning, and you can sort of take these forward and do sort of 10 by 5-fold cross validation on them. The models that we find in there, we look in the test fold at the correlation between those the levels that we predict and the levels that they actually are in that test fold. And then we do that sort of R squared is one of our metrics for the accuracy of the model. And then we also compute a similar sort of metric, but in external validation as well. And so those are our two sort of measures of success. Oops. So here, I'll just give you a sense of the accuracy of these models. So how predictable genetically are proteins, so as measured by the SomaScan platform. And it turns out that actually there's a subset that are quite predictable. So here you can see on the top left a kind of scatter plot of the internal test Spearman rank correlation versus the external validation Spearman rank correlation. You'd hope that those two things are fairly similar, that they all lie along the x equals y axis. And largely, that's what we see. There's some variation there. There's some, internally, these things seem to be working, but when you really take it out and do a really rigorous external validation, you get a decline in R squared. That happens for some proteins, to be expected. That can be for all sorts of reasons. But largely, you see a correlation there between the two correlations, which is good. And then on the top right, you can see the two sort of concentric circles comparing the kind of the rough sort of distribution of the variance explained, sort of internal validation on the outside and external validation on the inside. And you can see that some of those R squareds are appreciable. You can explain roughly 10 to 30% of the data using genetics for about 25% of the proteins, or 10% or more for about 25% of the proteins in external validation, which is pretty good. That R squared can also boost up. There's a small set of proteins there where you can explain half or more of the variation using genetics. And some of these are shown here on some of these proteins here are shown at the bottom here, sort of plotting out the variance, external validation variance explained, comparing interval and Fenlon there in the yellow. So very similar bars there, which is reassuring. Some of which you see a decline. So for example, this AAGAB, where you get kind of like sort of half of the variance explained in interval, but like, 5% or something like that, or less explained in Finland. We've looked into these specifically. Largely that's because the genetic variants that have that explain a lot of the variation aren't present in the external validation cohort. So you don't have that information to actually give you that boost.
1 minute, Mike.
Okay, that's good. So we see sort of similar patterns there for another proteomics platform. So Olink, similar sort of thing. And then importantly for Metabolon, we see that for different sets of individuals within the interval cohort, sort of roughly 10,000 and 10,000, we see very strong correlation. So a lot of these metabolites are, we can predict their levels from genetics pretty strongly. So we can also look at the pathway level, what percentage of proteins within particular pathways are kind of genetically predictable and how much. And you can see differences in these pathways. So for example, roughly 10%, 8 or 9, 10% or so of pathways across all of Reactome, which is a database of pathways, have, at least 1% of the variance explained in the proteins as explained by the genetics. And that can be, that can be quite high. So for example, for digestion and absorption, you get much more, maybe a third of the proteins in that super pathway, you've got some level of genetic prediction, sometimes quite strong. So interesting to look at these things. There's lots of interpretation now of our results and looking at them at the pathway level, also checking additional robustness, adding in additional types of biomolecules, that sort of thing. So that's how we're extending and deepening what we hope will be a resource, what we're calling an atlas of kind of genetic models that can help people predict their biomolecular traits in not just the cohorts we're looking at, but any cohort as well, right across the Multi-omics Consortium. So I'll end there. Hopefully that was fairly understandable in the small time that we had, and I'll hand it back to Adam.
Thanks very much, Mike. Really excellent start and a really great example of how combining data from multiple cohorts is important. In fact, you need, as you've shown, multiple cohorts here to be able to train the model and then to test how well it works. And I think you gave some nice example of both consistency between cohorts, but also differences between cohorts as well. In terms of a question, maybe I could start with asking, you showed that for some analytes, there's very kind of high proportion that's explained by the genetics. But how useful do you think those values are? So for example, is 30% of the variance explained by genetics sufficient to do something useful in another cohort, or 10%, or where would you say is enough to be useful?
Yeah, exactly. Look, it depends on what you want to, so is that level fit for purpose, right? It just depends on what you want to do. A common application of this is to use the predicted values, which are completely unmeasured in a separate cohort, and to look at the correlation between those predicted values and some outcome of interest, like incident coronary heart disease, incident diabetes, that sort of thing. It helps you identify biomarkers in a pretty, potentially efficient fashion, right? You don't have to go in and do the assay. You can look at it in silico if you've got the genotypes there. And there, it really depends on the level of that correlation, right? And that affects your power of detection, roughly in proportion to, you'd end up, there's a relationship between that R squared and the sample size in which you'd need to detect a certain association with a given strength. So it really depends. You'd be able to, if you had more than half the variance explained in protein X, and you had a strong association with an incident disease, I would say there's a fairly good chance that you could detect that association in another cohort, if the size is enough, that sort of thing. So it's a complex question, but we think that there's a fair few things that you can do, at least with that sort of top level, most genetically predictable biomolecules. Right,
that may be very efficient then, as you say, compared to spending millions of pounds in doing the actual measurements in several hundred thousand people. So that could be a very, very useful method, I think. A question from Jim Wilson. He asked about the comparison of the O-link and the SomaLogic you showed on 2 slides. Does one look better than the other?
Oh, I wouldn't venture to say better or worse at this stage. I think it's hard.
Can we have a quick look? Can we have a quick look at the slide that was covered so quickly?
Sure. Sorry, there we go. Back into it. So really, I mean, you're kind of looking at these concentric circles, and hopefully seeing more red or green than blue. And are these matching up? Yeah, no, the colors are a little up. Sorry, Jim, the answer to your question is not immediately apparent from the visualization that we've got, but maybe we can finish that up with you offline. I understand the interest in that question.
Yeah, that's why I asked the question, because of your fancy colors. I'd love to hear more about it. Thank you.
Yeah, we'll follow up with you on that for sure.
Thanks, and we may hear more about comparison between different assay platforms later in the day, I think. There's a couple more questions in the chat, but I think in the interest of time, we really need to move on. So Mike, if you wouldn't mind, if you're able to hang around to answering those in the chat function, that would be helpful.
Will do.
So we'll move on now. The next speaker, delighted, has joined us is Michael Holmes. He's an associate professor at the Nuffield Department of Public Health at the University of Oxford, and is a lead investigator in the China Kadoorie Biobank, which is not obviously a UK population cohort, but is part led from the UK and has, again, the kinds of data we talked about at the start in terms of genetics, some multi-omics and health records. So it's an obvious source of potential international collaboration and replication. So welcome, Michael. Look forward to hearing your talk.
Thanks, Adam. Thanks, everyone, for the opportunity to speak today. So yeah, so I'm going to talk about some omics applications to the China Kadoorie Biobank. Just to check, can you hear and see my slide?
Yep, that's all great.
Brilliant, thanks. So let's see if I can navigate. Okay, so most of you, well, many people will be familiar with this China Kadoorie Biobank as a sister study to the UK Biobank. And we have just over 500,000 individuals selected from 10 different geographic regions in China, spanning urban, rural regions and areas with very different weather systems, providing very diverse environmental heterogeneity, in addition to the ancestral heterogeneity to Europeans, which are conventionally analysed in large biobanks, so providing a really rich opportunity to explore disease associations, and in some ways it's the heterogeneity between populations that are diverse that can help us to triangulate to important new discoveries. So just as a sort of description of what we have available in CKB, in all 500,000, we have information at baseline on for example, smoking, alcohol, diet. We also have phenotyping, including blood pressure, measures of adiposity, heart rate, height and weight, and so forth. And every few years, we have resurveys in a subset, which allows us to conduct repeat measurements for regression, dilution, and corrections, but also conduct de novo phenotyping, including, for example, bone mineral density, hand grip strength, and carotid ultrasound phenotyping, and so forth. In varying numbers of individuals, we have processed samples. So everybody, all individuals provided blood samples at baseline, and in a subset of individuals, we have, for example, GWAS genotyping in 100,000 individuals, blood biochemistry in roughly 20,000 or so, and proteomics, metabolomics in further subsets, as I'm going to describe. And clearly, the value is in the incident events that occur, which we capture through electronic health records and death and disease registries. And I think these numbers are slightly out of date, but we have over 40,000 deaths and over 1 million hospitalizations, including over 1,300 disease types. So today I'm going to be talking about sort of showcasing some of the work that we've done in measuring proteomics and metabolomics in the CKB. But I just wanted to use this slide to illustrate that rather than looking at these in isolation, I think the real value is in looking at these traits in terms of pathways from genotype and also clearly the environment through to disease. So these don't exist in isolation, but rather are parts of pathways to disease. And this figure is taken from a recent review, which we published in Nature Reviews Cardiology, which just describes some of the applications of genomics, including proteomics, genomics and downstream omics, including proteomics to drug discovery. So like I said, I'm going to focus on applications of proteomics and metabolomics, and in describing the phenotyping we have available. So we currently have proteomics measured through one of the Olink 92 panels, which is the one we've used so far is the immuno-oncology one, funded through Pancreatic Cancer UK and CUK, which measures 92 proteins, and we have this measured in 1,400 individuals, which is a relatively small subset, but that includes 700 incident cases of pancreatic cancer and 700 population-based controls. In addition, we're going to be measuring very soon additional Olink proteomics in roughly 2,000 adjudicated incident MI cases and 2,000 controls, and that will provide us with the opportunity to triangulate the observational associations with the MR associations in East Europe, in East Asians, and then potentially the opportunity for transethnic GWAS and transethnic Mendelian randomization analysis, which I think is really important, especially when we think about developing medicines which these proteins may represent as in therapeutic targets, when we think about developing them for not only for European populations, but for non-European individuals, so that we have medicines that we can demonstrate through human genetics that are likely to lead to benefits to human health. And then in metabolomics field, we have the NMR Metabolomics platform quantified through the BrainShake or Nightingale Health assay, and that's in 10,000 individuals, which consists of individuals with incident CVD, diabetes, cancer, and population-based controls. And then in a smaller subset, we have the Metabolon HD4 array, which is in 400 individuals, including 200 incident cases of non-alcoholic fatty liver disease on AFOD. So one of the interests that we have, and I think many of us have, is can we use these omics assays to try to start to tease apart disease pathways from complex exposures such as body mass index to disease? And this might be potentially lucrative, because if we can identify potential intermediaries on these pathways, and if those intermediaries are druggable, then we might identify mechanisms by which we can offset the harm that arises from excess adiposity, and clearly, ideally, we would lower our BMIs or wasted ratios, but as I think it's appreciable, that's actually very hard to do both at an individual and at a population level. So while we tackle with that conundrum of how to modify these complex biomarkers, can we use MR and observational studies to tease apart these causal pathways as a means to develop potential new treatment opportunities? And so this is work which we did in the China Could Review Biobank. This is the immuno-oncology panel, and this is in the 600 individuals that are population-based controls. So pretty small sample sizes when we think about what we currently typically do with large-scale epidemiology. But I guess this shows the power of biobanks and proteomics and the assays that are available now, which are really quite reliable. And so this is a subset of these 92 proteins which are observationally associated to be correlated with BMI. So these are cross-sectional associations, and clearly observational analyses, as we all are familiar with, can be biased or have errors in them due to reverse causation or confounding. And so we can then reappraise them through genetic approaches such as Mendelian randomization, and when we then reevaluate these relationships through MR, and again, this is still in the 600 individuals, again showing the value of what can be done in even small sample sizes. We can see that, for example, is congruence of findings of BMI with higher concentrations of interleukin-6 and interleukin-18. But interesting, also potentially important divergence where the MR estimate is directionally opposite to the observational estimates, and that can itself highlight important confounding pathways. But the other message here is that we do see the sort of rich tapestry of causation. So it's not that, okay, BMI only associates with 1 or 2 proteins. Remember, this is 92 proteins that are selected on the basis of their potential role in immunology and oncology. So if we could expand that to across the proteome, then this probably heralds a large effect of adiposity on the proteome. But the point being here, going back to this slide here, we can begin to sort of characterize the observational association of BMI with these proteins, and then interrogate whether those relationships are likely to be cause and effect. And then clearly the next important step to dissecting the disease pathways is asking, are these proteins then going on to lead to alterations and risk of disease? And taking this forward again in the China-Korea Biobank, again in this relatively small sample size. So of the 600 population-based individuals, 150 had incident cardiovascular disease, and that's, I mean, again, these are small numbers, which is why the 95% confidence intervals are relatively wide. But again, you can begin to tease apart patterns of associations and really begin to start thinking about, okay, so we have this information from BMI to proteins, information from proteomics to disease. Are these tractable therapeutic targets? And clearly, what we need now is genetic instruments which reliably allow us to conduct MR analyses using cis-pQTLs. And that's where endeavors such as the Scallop Consortium and other initiatives will be incredibly valuable. So what is the added value of omics in this context? Well, the potential identification of proteins that might mediate the harmful effects of excess adiposity and risk of cardiovascular disease, and the importance being that such proteins may represent previously identified etiological pathways and potential therapeutic targets. So moving track downstream from the proteins to the metabolites, and just sort of showcasing some work which we published a couple of years ago in NMR Metabolomics. So this is published in JACC, where we compared and contrasted the observational associations of lipoprotein, lipids, and other traits quantified on the NMR assay with incident MI, incident ischemic stroke, and incident intracerebral hemorrhage. And what this figure, excuse me, on the left shows is the associations of these traits across these 3 cardiovascular disease types. So each blue blob is an individual metabolite, and on the x-axis here is, for example, the association with incident ischemic stroke, and the y-axis is the association with MI. And the associations, I mean, obviously there is some noise here, but the associations tend to be similar in direction, and suggesting that there are potential these lipid lipoprotein traits, with all the nuances of interpretation of observational epidemiology, that ischemic stroke and MI might have shared etiological pathways specifically from these lipid entities. And interesting, when you swap out on the y-axis MI for intracerebral hemorrhage, we don't see that, and we see that these lipoprotein lipids and their related traits have much weaker associations with intracerebral hemorrhage, suggesting divergence of association. And when we look at the sort of cartoon plot on the right-hand side here, we can see the triglyceride and cholesterol concentration of the ApoB containing lipoprotein, so very low density lipoprotein, intermediate and low density lipoprotein. And we see here that the triglycerides and the cholesterol in these lipoprotein subclasses are typically positively associated with MI and ischemic stroke, but not with ICH, as we saw on the left-hand side here. But interestingly, when you get down to HDL, the cholesterol is inversely associated, as we would expect from previous studies done by, for example, Emerging Risk Factor Consortium. But we see a divergence here where the cholesterol is inversely associated with the triglycerides is positively associated. And this potentially speaks to important features of HDL metabolism, which might have relevance for disease. And obviously that needs picking apart, and future endeavors, and for example, the UK Biobank with much larger phenotyping, providing opportunities for genetic instrument derivation might be helpful here in asking, for example, does the I think everyone's familiar with the concept that cholesterol and HDL is unlikely to be causally related to heart disease, but potentially there's a role of drugs that modify triglyceride concentrations as a discrete mechanism to exploiting lipid alterations in HDL as an opportunity for thinking about other ways to lower heart disease risk. So
1 minute, Mike.
Oh gosh, okay, great. Sorry about that. The other thing is that while there was this divergence or this lack of association of lipids with intracerebral hemorrhage, we did see congruent associations of glycoprotein acetals, which is an inflammation marker, across these ischemic and hemorrhagic disease types, which potentially, and I think where you see the divergence and the congruence, that's where you can start to tease apart potential meaning here in terms of etiology. So being very quick, the added value is the detailed exploration of lipoprotein lipids, the insight of the overlap of lipid pathways for atherosclerotic CVD in contradistinction to intracerebral hemorrhage, that there's this divergence of triglycerides versus HDL triglycerides versus cholesterol and HDL species, and a potential shared role of inflammation in MI, ischemic stroke, and intracerebral hemorrhage. We've got a couple of slides left. I'll just showcase some work which we're doing now, looking at fatty liver disease. And this is not your typical Manhattan plot of GWAS, but these are, each blob here is an individual metabolite, and these are associations with body mass index, waist circumference, and non-alcoholic fatty liver disease, and the color coding is by domain of these metabolites. And again, we can start to think about, okay, so here, for example, here we have glutamate strongly associated with body mass index, waist circumference, and NAFLD. And I'm aware this is a very superficial exploration of what these data can do. But in terms of what it can do, the metabolome provides, I think, a much broader perspective of the metabolome as compared to the NMR platform, allowing potential hypothesis-free explorations of metabolomic associations with adiposity and NAFLD. And again, as we saw with the BMI to heart disease, we can start thinking about, okay, so I think it's not controversial to say that BMI causes NAFLD, and therefore can we think about these metabolites as being on a causal pathway? And while metabolites themselves are probably very hard to modify therapeutically, we can then use the genetic architecture of metabolites to think about what proteins modify those metabolites, and are those proteins then potential therapeutic modalities to preventing the disease burden that arises from NAFLD. So the conclusions are that omics provides opportunities to discover novel disease associations, clarifying etiological pathways, can shed light on potential therapeutic targets. And I'm really excited that we're going to be measuring these additional proteins in 2,000 incident MI cases and 2,000 controls, and really keen and hopeful that this will provide meaningful new discoveries that potentially translate into medicines. And just to finish by thanking the CKB participants, the scientific teams at Oxford and China, Huan Jipang, who was a PhD student at Oxford, who's now a postdoc at Peking University, who did a lot of these data analyses with Christiana Karcznacki, who's a statistician at Oxford, Simon Chen, who's the PI of CKB, and our funders, and a special acknowledgement to BHF that funds me through a personal fellowship. Thanks very much, everyone. Happy to take any questions.
Brilliant. Thanks, Michael. Fantastic overview there of how you can do exactly what we said at the start, integrate genetic data, multi-omic data, disease information, this time in a single large biobank, but to provide some really important insights into causes of different diseases. I guess an obvious question is how easy you think it would be to compare or integrate findings from this large Chinese biobank with findings from UK cohorts.
So, I mean, there's a lot of methodological development going on into things, into transethnic GWAS, and we're certainly in CKB, we're focusing on that, for example, at the moment. We've done quite a lot of work in adiposity GWAS, and there's a lot of work being done in sort of portability of genetic instruments, and certainly that will play a valuable role in then thinking about how can we then conduct, can we use transectic GWAS to better select instruments? Because, for example, what we've seen so far, I think it's correct to summarize, is that if you do a GWAS of BMI in China Kudruvi Biobank and then do a transethnic GWAS of BMI and CKB and UKB, then actually that transethnic instrument performs better in CKB. So I think there's a lot of synergistic opportunities there, in addition to just the simple or the qualitative or quantitative comparisons of, for example, observational associations in these diverse biobanks. But as I said before as well, I think the real advantage of CKB in many respects is the environmental heterogeneity as well, which provides us with opportunities to explore things in ways and triangulate the different sources of information to hopefully provide additional dimensions of information.
Thanks. I think I agree with all of that. Do please continue to put any questions into the chat, and Michael may have some time to answer them before he goes off to help with the vaccination program this afternoon. So thanks for the great talk, Michael, and look forward to engaging on ways we can collaborate more in future.
Thanks, Adam. Thanks, everyone.
We're going to move on now to the third in a series of Michaels. Mike Pietzner, who's a senior research associate at the MRC Epidemiology Unit here in Cambridge and has been working a lot with the Fenland and Epic Norfolk cohorts and thinking a lot about cross-platform approaches. So Mike, welcome and over to you.
Thank you, Adam. And thank you all for your interest in our work. So I'm here to present some of our work crossing the platform, so basically integrating different technologies and the hurdles and successes we have made so far. So, yes. So you have heard about the omics, and I just want to give a brief reminder. So what we're basically talking about is the flow of biological information. So you all know that our genes, this is what we are born with. And then, there are different entities which actually make it happen, that what we have encoded in the gene gets into life, basically happening at this side. So the proteins facilitate life, and the metabolism is which gives energy. So like the burning of glucose to generate energy, and these are the two entities I'm going to talk about today. So the metabolome covers a wide variety of small molecules, basically only defined by their size. And one of the things all of you might be familiar with is glucose. You may have already measured your blood glucose or blood sugar, but there are also metabolites which have the biological activity, like hormones, like cortisol, and also molecules that are covered by dietary intake, for instance, polyunsaturated fatty acids. And there are only two distinct techniques to measure the metabolome. They have been mentioned already in talks from the other two mics. So there's mass spectrometry, which is basically weighing your metabolites and distinguish based on the weight what metabolite it might be. And there's nuclear magnetic resonance spectroscopy, which basically works similar to an MRI, which uses the biophysical properties of metabolites to distinguish and see what's in your blood sample. And they give a slightly different coverage, and mass spec is actually the one having the higher coverage, as Mike just mentioned. So the first thing is, why are we interested in those metabolites? And that's why we did a fairly comprehensive study in the cohort called Epic Norfolk, which covers around 1,000 metabolites. So we've measured those at baseline examinations, and then we followed up those around 11,000 participants using EHR data. So we asked the hospitals to give us records whether any of those participants developed any sort of disease, and we looked at 27 diseases. And we wanted to know whether the level of a metabolite at baseline has somehow predictive value for what happens in the future. And what we did see is that almost half of the metabolites were associated with at least one of those diseases, meaning that a higher level of this metabolite at baseline was associated with a higher risk, or vice versa, like lower levels were associated with lower risk. And the astonishing thing was that these effects, and I think Mike touched on this already, are not happening in isolation. So 2/3 of those associations were actually shared among at least two diseases. And this is what this chart will tell you. So for each outcome, for instance, COPD, we see the number of metabolites, which is like proportional to the bar chart. So we have many metabolites associated with COPD, less so with some cancer outcomes. But what we do see is there's a colored fraction. And actually the colored fraction is this what connects diseases across each other. So because this indicates that a metabolite, or maybe hundreds, tens or hundreds of those, are shared with at least one other disease. And you do see the connection is like all over the board. So these could be seen as a antecedents of multimorbidity, meaning that people do not develop just type 2 diabetes or just have a heart attack. Often things occur in people, in the same people. So meaning that these metabolites might help us to understand the increasing multimorbidity burden. And just to give a brief example, for instance, we saw that a carbohydrate called N-acetylneuraminate was associated with the onset of 14 diseases across the board, including cancers, including type 2 diabetes. So there's diverse and very meaningful biological information in those data. However, there's always the question whether what we see here in these observational studies might be due to a factor we haven't accounted for yet, meaning we may have not measured the real underlying cause, and this is what we usually worry about. And that's why we go a step further to utilize genetics. And so what we did is we tried to aggregate information on a defined set of around 180 metabolites, which are commonly detected by 4 different technologies. And this is what is displayed here in these colorful bars. So for instance, tyrosine is covered by a kit called from a company Biomolecules and Metabolomic, which both use MS or mass spectrometry. Then it's also covered by Nightingale, which uses NMR. And the slightly different, there are also differences in those methods between Metabolomic and Biomolecules actually. What we could see is that the effect anchored on a genome, so like the common genetic variation, which makes the difference among us, has a strong effect on metabolites, but these effects are well captured by each of the platforms, and we observe little evidence of heterogeneity across those platforms. So we could say that the genetic effects are conserved across these platforms, and we were actually able to, by having this immense increase in sample size, to establish a causal role of stereo metabolism in a rare eye disorder. And this opened up opportunities to think about supplementation strategies, because obviously you could start maybe recommending a trial which looks into whether serine supplementation may attenuate the risk or mitigate it. So this is what we have done so far, but obviously our work package is based on the proteome. And maybe many of you, if they hear the word protein, they think about the protein content in your daily food intake. But I want to make it a bit more broad, basically introducing proteins as a chain of amino acids, which can fold in really sophisticated 3-dimensional structures and then facilitate all these biological important functions. For instance, this is an example of hemoglobin, which is basically the protein which facilitates the transport of oxygen in your blood. So they have a multitude of functions, and it's why we are so interested in what determines those levels. So there are a couple of ways, and my previous speakers already started introducing those. So just for the sake of completeness, so one could again use mass spectrometry, and what you do is you take your proteins, cut them into little pieces, and then weigh each little piece, and then you basically use some fancy algorithms to reassemble those in the computer to give you protein levels. However, this is until now very restricted in the sense that it covers not that many proteins. And then that's where these 2 technologies came in, which is basically what is behind Olink. It's called proximity extension assay. What they use is a technique which is fairly established in the sense of that we use antibodies in clinical chemistry for decades to detect protein levels in your blood. And many clinical tests are based on antibodies in the clinic. And then there's a company called SomaLogic, which developed these aptamer technologies. These are little fragments of actually also DNA, which fits like a key into a lock to a certain protein, and then you can basically fish out those proteins and see how much you get in an array design. So we're obviously interested how good compare those platforms, particularly those who have a broad coverage. And what we did is we measured many of those, or thousands of those proteins in the Finland cohort, including roughly 12,000 participants, for which we have measured for roughly 5,000 proteins, which is a lot. And then we have a subsample of this study covering around 500 participants, where we also have these different technique. And you see here the type of how the protein, which is this green little thingy here, is targeted is fairly different. So both attempts might give also different results. And what we have basically is for around 870 proteins, we have measurements using SomoScan, and we have measurements based on O-link in the same participant. So what we can do is we can look how good do both compare. And we do see excellent agreement. This is a protein called leptin, which is involved in appetite regulation. And you do see what is measured by SomoScan nicely aligns with what is measured by O-link, as seen on most of the points fall on this diagonal. However, we also see proteins where we do not see any relation. So it's basically a random crowd of noise. And more weirdly, we also see inverse relations, meaning that high levels in the SomoScan assay give low levels in the O-link assay. So we have a wide variety of variation. Again, these are all supposed to target the exact same protein, but actually what we measure or how we measure matters. So then we set off to investigate whether this has any biological relevance, and we did so using a genetic anchor. So these are what is called Manhattan-like plots. So each of these high peaks would indicate there is a genetic variant which strongly affects the protein level. So whether or not you have this genetic variation, you may have a higher or lower protein level. And then we wanted to investigate whether the local, like, is this the exact same genetic variant, or is it just close by? And we also wanted to understand whether the effect direction. So is it a consistent effect? So both assays have a positive association with the genetic variant, or might it be that only one assay is related to the genetic variant, or is it even the case that the genetic variant is the same, but the effect are opposing, similar as I just showed you? And all of this is, yeah, nice to know in a sense, but the question is there any biological relevance? And then what we also went for is we integrated genetic signals where we know that they relate to disease onset or they influence gene expression, meaning basically the template for those proteins. Is there more or less of those in certain tissues? And in a nutshell, what we do see is by leveraging machine learning, we identify a couple of characteristics that seem to influence what both assays measure. And this is like a cartoon summarizing those. And we do see effects related to the protein, whether or not this has what is called the transmembrane domain, meaning that there is a part of the protein which resides in the cell membrane. But we also see effects of the genetic variant itself. So whether on, it's 1 minute, Adam?
Yes, please. Thank you.
Sure. So quite a lot of factors which influence, but also your genetic background seems to influence what we measure. However, we see that around 2/3 of the genetic findings are consistent. We observe some not worrying, but contradicting results, for instance, for a protein called PILRA and Alzheimer's disease, where effect directions do not align. But we also try to provide guidance how to incorporate different layers of information to get around this. So basically, what we've learned so far is that integration of these platforms is essential to maximize power, to generate cohort reproducibility across cohorts, but all come with its own challenges. We do have to understand them before we can naively combine studies, meaning we would otherwise lose or signals or get false positives hit. And it's important to do this in a systematic and bidirectional comparison to see what differs and what might be consistent. And in particular for the genetic discovery for protein studies, it's not only that we have this immense gain in looking at disease mechanisms, but it also seems to affect how we measure the proteins in the first place, which is important for finding. And obviously, all this is not done by me alone. This is a huge team effort with Claudia and Varun leading this work package. And with that, I'd like to thank you for your attention.
Brilliant, thanks, Mike. Fantastic talk, and I think really illustrates some of the key features of the consortium in terms of multi-cohort analyses, but also one of the key challenges that reviewers pushed us on when we applied for this funding about how can we integrate across different platforms. And I think you nicely showed there how for Metabolomics, maybe the integration is more straightforward and worked very well, whereas for proteomics, there are sometimes some that work very well, but also some challenges. How do you think we can learn from integrating across these different platforms in terms of working out what the real biology is there? So for example, if you have conflicting information from different platforms, how do you think we should handle that?
I mean, what we have seen and when we try to investigate factors which might give a better handle on what is consistent or not is that integration of gene expression or integration of phenotypic information. So is the genetic variance we see somehow related to a phenotype? This could be a biomarker, this could be a disease endpoint. This can help to increase our credibility that this, or increase the credibility that this is a true PQTL, if you so want. But what we are actually lacking is some sort of gold standard where we can all calibrate this against. But you can imagine that having 5,000 proteins measured by a gold standard is simply impossible. So that's why we try to leverage all these types of information to get more confident about how we follow up and what we may miss, or get at least an idea of what we may miss.
Thanks, and I think that's really important because so much of the science we do has been aggregated around particular platforms, and we've looked at one platform at a time, and I think you're really pioneering methods that look at multiple platforms and get a much deeper insight of what we're really measuring and what the implications are. So great work, and thanks for such a clear talk. So finally this morning we're going to hear from Professor Joanna Zulaki, who's a professor of chronic disease epidemiology at Imperial College in London. Over to you, Joanna.
Hi, Adam. Thanks very much. I'm sharing my screen. Here we are. I hope you can all see this. Sorry, I wasn't ready.
Yeah, we can see that. I don't know if you want to go to the, yeah, there you go.
All right, so my talk again is on metabolomics and genomics. I sort of anticipated the previous talk, so maybe I have a very short introduction on metabolomics that until now we've covered. That with this method, we try to get a very broad picture of the small molecules in the biological fluid, usually blood, which give us ideas on the pathways and the metabolic networks that work, they are activated in the body, and we try to link those with diseases and other phenotypes. So we have two main technologies, the NMR and the mass spec that Mike also mentioned. And unlike genomics, where we have a single technology to measure them and one genome, with the metabolomics it's a little bit more complicated, as different studies use different platforms, different ways to analyze metabolomics, sometimes different fluids as well, which makes the integration across cohorts difficult. We have worked on that, so we have worked on workflows and pipelines on how to bring together either NMR data or mass spectrometry data across cohorts, focusing on untargeted data, which means that we have we measure as many metabolites as possible in blood or urine without worrying at the annotation at the beginning, without knowing the chemical identity of the metabolites. But we do that subsequently when we know which of them is important for us. That has benefits because we don't have an a priori hypothesis on what we think it's important, but also it has limitations in terms of the translation of the data to pathways, because sometimes we are unable to identify the metabolite that we are interested in. But the pipeline is looking like this, where we try to align different cohorts or match the masses between the mass spectrometry across cohorts, and then we lead to the data analysis and interpretation. And we have successfully done that for NMR. For example, here it's 2 years ago, and I have presented that before, so I won't go into detail, but it's an application of linking NMR data across 3 different cohorts in order to find associations with subclinical atherosclerosis measured in the carotid and coronary arteries. So we took that step further in trying to bring together mass spectrometry data, the MS data, which is far more challenging when we have it in untargeted mode. And for example, here, each of these 3 cohorts have a different sample size, which is okay, but they also have different dimensions of metabolic features that are measured each time. So in order to match them, we need to come to a one size cohort, which sort of matches together these features that are common between the 3 cohorts. And to do that, we have worked on, again, workflows for this analysis. And this is, methodological work from Rui Pinto, who is a chemometrician. And he has developed, workflows on how to find one peak in one data set, and then define a window around this peak, a window in terms of the mass and the retention time that are important in the MS results, and how to find what is the equivalent of that dot to the other cohorts and the other data sets in order to match these two and bring them together. So I'm not going to go into details on how this works, but we are trying to build that methodologies in order to link untargeted data sets together and increase our power and analyze them together. So now we are working on the matched cohorts in terms of the subclinical atherosclerosis, and we are preparing the results on that. However, going a step further this time, we are also interested to explain the pathways of the metabolomics in terms of linking with other omics, and this time with genetics. So we have a plethora of genome-wide association studies, and we know a lot on the genes and the variation of the genome that is associated with different phenotypes and different diseases. What we don't know is that many of these findings, how exactly this genetic variation is linked to the phenotype of interest, remains unknown for many of those discoveries. And that is limiting the translational potential of the GWAS findings. So instead of going, as we heard before, from the metabolites to the genetic predisposition to the metabolites, we took a different approach where we went from the GWAS findings, so the genetic variation that we know it is important for disease, to the metabolomics in order to find whether this variation is associated with particular metabolic pathways associated with these SNPs, with the genetic variation, and subsequently whether this variation is associated with a disease. So whether this could be horizontal or vertical pleiotropy, because we would be interested to the horizontal one where we go from the genetics to the metabolic pathways to the phenotypes. And this process will, again, as before, allow us to depict those pathways that are important for the diseases and could help us explain those findings and give us a clearer picture. So I'll give an example. We are doing this in different diseases. So we have a large project on cardiovascular disease, colorectal cancer, and this is Alzheimer's disease, which I chose to go deeper today. That's work led by Abbas, where we took the Alzheimer's disease GWAS hits. So we know around 47, if I'm not mistaken, separate SNPs that are associated with Alzheimer's disease, genetic variation. For very few of those, the ones only that are familiar, that belong to familiar clustering and early onset Alzheimer's, we know the mechanism that may lead to Alzheimer's disease. For the vast majority of those, we don't know the molecular mechanism. So we thought we can associate the genetic variation with metabolomics data, and then follow up this with a series of experiments to support causality, and also with experimental validation in lab studies and animal studies. And this is our more focused workflow of that work. So we had two cohorts this time with metabolomics data, that they have different types of metabolomics data. They both have MS from the National Phenome Center at Imperial, which is untargeted MS data, and also Metabolon. So we have different elements of metabolomics data. This is lipid positive, it's an assay on MS data, lipid negative and helic positive plus metabolon. And we also know from the literature the genetic variation, the SNPs that have been associated with Alzheimer's disease. But what we did is that we did what we call an MWAS, a metabolite-wide association study for each of those SNPs. And we found interesting results for particular SNPs belonging to the APCA7 gene. I'm going to show that in the next slide, and followed this up with experimental validation in animal studies, with Mendelian randomization to support causality, and with associations with phenotypes. So that could be an electronic health record association where we had a lot of outcomes. In this particular case, we had cognitive function as an intermediate phenotype to Alzheimer's disease. So this is a flavor of the results. Here we see, it might be a little bit small, but we see the different 48 SNPs that we know that are important for Alzheimer's. And here we have focused on these particular results in the APCA7 gene, where we had a lot of associations, which we then annotated from the Phenome Center, all within the lactoceramide pathway. So we had a number of associations here. We had a few others, but we concentrated on this one because we had most of the associations, and all belonging to ceramides and lactoceramides that belong to the same pathway. Here are the different SNPs within the APCA7 gene that showed associations. So all of those SNPs showed associations. And we also noticed that some of those associations with the lactoceramides are also associations found from the ApoE and two SNPs within the ApoE gene with the same metabolite. So in effect, that gave us the link between metabolomic pathways connected with the APCA7 and the ApoE gene for Alzheimer's disease. And you see here we have, we were able to get the same metabolites from different platforms. So again, giving internal validity to the results were the same metabolites found from Metabolon and from the National Phenome Center assays. Moving forward, we also looked at associations between this group of metabolites that were associated with the genes, in the SNPs in the APCA7, with a finger trial. This is a trial on people at high risk of developing Alzheimer's because they had low cognitive score for their age. So we had data on metabolomics there, and we sort of found 3 associations between the 3 main lactoceramides that we were interested in, giving lower cognitive scores. Unfortunately, those were not supported in the airwave cohort, but that's a cohort where the median age is 40. They are young people, and maybe so in that early age, we cannot find yet associations with cognitive function score. Importantly, we also did Mendelian randomizations with those lactoceramides that were of interest, and we were able to support causality with 3 of them, with sensitivity analysis supporting the main analysis. We see the MR here is not significant, but it is consistent, and we didn't have high heterogeneity. Now, all this work has been supported with a line of experiments in the lab, which I haven't gone deep today because it's a little bit out of the scope, but we tested directly for an association between concentrations of ceramide species and APCA7, carrying out metabolic profiling of brain tissue from the cortex of knockout mouse with knockout of APCA7 versus wild type to test whether the lipidomic variation related to APCA7 in peripheral blood in humans were also found in the brain tissue. And this data also supported the link between the APCA7 and regulation of ceramide metabolism in the brain. So we came up with a proposed interaction between the ceramide metabolism and APCA7 in the brain. The figure here shows the proposed picture, but in summary, we propose that, in addition to regulating cholesterol, APCA7 also regulates intracellular concentrations of ceramides and exocyl ceramides, which are also found to be increasing the cortex of mice. So we hypothesize that the accumulation of these ceramide species in the brain could increase the cleavage of APP, which further induce plaque formation, as has been previously suggested by other studies linking to formation of plaques linked to Alzheimer's disease. So having such a picture, the same picture coming from different lines of evidence through linking genomics to metabolomics, but also going back to observational data, supporting the hypothesis with Mendelian randomization and with basic science experiments. I think we can bring more confidence to the results and going deeper into molecular pathways linking genes to diseases. So the integration here of genomics and metabolomics across human and animal studies provided biological insights, and then further this can provide novel targets for this prevention and treatment of the disease. And that was a collaborative project with Abbas, Paul Elliott, Abbas Denhan, and Julian Griffin from the Metabolomic sides, and Rui Pinto and Ibrahim Karman were our 2 postdocs that carried out most of the work that I presented today.
Fantastic. Thanks very much, Joanna. I think there's a really nice example, as you say, of how we can integrate genomic and metabolomics data in humans, but then also support our findings by other types of evidence as well, in this case, studies in mice. And I think you also highlight one of the potential opportunities here, which is when sometimes we don't have quite the right kind of cohort to replicate our results. You had, in that case, your participants in Airwave were probably, as you say, too young to have many people who'd gone on to develop Alzheimer's disease, but maybe then expanding to see which other cohorts might have more cases, and importantly, the same kind of measures that you have to enable a measurement of the same kinds of metabolites. So I think that kind of highlights the opportunities we have here as well to use this community to further support our results.
The need to bring data together, to have the electronic health record as well, to be able to validate those findings.
Exactly, exactly. Thank you. That was a great talk and a really great way to finish an excellent morning. So thanks to Mike, and Joanna for fantastic talks. They've really, I think, showcased the value of the data and the community that we have here. We're going to take a break now for lunch because I think it's been a lot of information. We've got 4 more exciting talks to come this afternoon in slightly different areas to what you've heard this morning. So thank you again to all the speakers and for keeping broadly to time. Do continue to put questions in the chat if you'd like people to answer them. And I think I saw some questions about our use of acronyms as well. So if there are phrases or words you don't understand, do feel free to put those into the chat and people can try and answer those for you. Just in terms of the logistics, if people do want to stay on the Zoom to avoid having to rejoin and go into the waiting room, that's fine. I suggest you leave your cameras and microphones off. Alternatively, we'll have a sweep at 12:45 promptly to let people in who are waiting, and we'll start again then with some more of the exciting science talks. So enjoy your lunch and see you all in about 20 minutes. Well, welcome back, everybody. We're at 12:45. Hope everyone got a good break, maybe even got a chance to get some fresh air. We've got some more exciting talks lined up for this afternoon. And to kick us off, we're going to hear first from Amanda Stranks, who leads the public and patient engagement and involvement section of the multi-omics program. And I think she's going to be helped by Sarah and Chris as well. So Amanda, over to you.
Absolutely, thanks Adam. And I have remembered to turn myself off mute, so that's a good start. Welcome everyone, thanks for coming back after lunch. It's really great to be able to have a talk about our PPI for this particular project. And I'm joined today by my co-lead Sarah, who's in the audience, and also Chris from our public panel. And just as an explanation of who we are, I am, as well as being co-lead on this particular project, I am the PPI lead at Cambridge Biomedical Research Centre and also on what feels like multiple other HDR UK projects. Similarly with Sarah, who is the lead for PPI at the Blood and Transplant Research Unit and also on the Understanding Causes of Disease project, and joined by our public member, Chris, who is on multiple committees as well, by the looks of things. And there are also many members of the public of our HDR UK Cambridge advisory group in the audience, and it's really nice to see some of those here as well. So what have we been doing for patient and public involvement for this particular project? Well, we've been very busy, which is great. And our advisory group have been absolutely instrumental in helping us to develop and contribute to many of the major grant applications that we've done through this particular project, as well as being playing a really important role in devising how the public group runs itself. So they've helped us to recruit new members, to put together the terms of reference, and to really help us understand how to best do public involvement for this project, which has otherwise been quite challenging. And then we've also got some public members on the steering committees of various smaller projects so that we have real public feedback at the centre of all of this work. Engagement-wise, we've done lots of educational seminars and workshops, as we sort of go on to describe later on. One of the real challenges of this project is helping members of the public understand what it is that we're doing with all of this data, and probably even more complex, what we mean by multi-omics, and even to a certain extent multi-morbidity. So there have been lots and lots of work that Sarah and I have been doing with the various work package leads to try and really convey what it is that we're trying to do and how people's data are being used. And then to support all of those, we have various newsletters and a voice on a website and through Twitter, just to make sure that we're always keeping people updated with how the project's developing. So the challenges of involving members of the public in this project are a few, I will say, starting with the fact that HDR UK itself is quite a complicated organisation. Even between Sarah and Chris and I, we cover multiple different projects. And again, just helping the public to understand what is already happening with health data and what we want to do with it is quite complex. And that sort of adds to things. I think there's also a certain not exactly a difficulty between funder and public needs, but they're not always the same. And being able to really ask members of the public what they want for this project and help it to align with the overall goals can sometimes be challenging, partly because just it's such a big project and there are so many different projects and people involved. Recruitment to our public group has not so much been challenging, but to make sure that it is the diverse group that we really would like it to be. And because there are so many cohorts from the various research projects that are taking part in this project, we want to make sure that we have members from each of the cohorts that are providing data into this project on our public group, diverse representation from across the UK geographically, and then also people from a range of backgrounds, be that ethnic backgrounds, educational backgrounds, and things like that. So trying to make sure that we really have a wide range of voices has been a little bit challenging, and is something that we've been working on. For Sarah and I particularly, it has been a big science communication challenge. I have a background in immunology and public health and molecular biology, and even I've sort of struggled at times to be able to talk clearly about what we're doing. And we've sort of seen some of our public members in the chat today sort of talk about the various acronyms and the challenging projects. So being able to really, again, just create that picture of what it is that we're trying to do and what are the tools and the technologies that we're using can be quite challenging. It's also been a bit difficult at times, or a bit challenging, just to balance that involvement and engagement. It's very easy to talk about what's going on. It's not always easy to really get public feedback that can change things to the same level as we would in other projects. And that's partly because health data itself is just a different kind of way of doing research. And many of the research projects that we're talking about have already taken place, in the sense that we're using data from existing cohorts, and that creates some challenges there. And then equally with patient and public involvement, trying to get the right balance between breadth, so that number of people that we're trying to involve, as I was saying, but also depth in terms of the depth of the relationships and the people that we can meet, and trying to find the right balance so that we're working really closely with some people, but also working with a wide range of people. And having said that, for all the talk about challenges, I know Sarah and I really enjoyed trying to come up with the best way to do patient and public involvement for this. And all of the members of our advisory group are absolutely instrumental. Their ideas and their feedback have been really, really valuable in improving everything that we do for this project. So what are we hoping to do with the sort of remainder of this project and leading into everything else? Well, to keep up recruitment. So as I keep up recruitment to our advisory committee and group. As I mentioned, we're looking for people right across the country, people that come from the cohorts that are providing data into this project, people living with multimorbidity, with all kinds of backgrounds. So if you do know anyone, all of the researchers who are watching, or even members of our advisory committee, if you know people who have a background that might be suitable to participate in this project, please let us know, because we're always looking for more members. We also need to provide more training opportunities for our members. So a lot of the stuff about multimorbidity that we've already covered, but things about the governance of health data, for example, about artificial intelligence, about data security, are all ideas that we want to cover for training options this year. And then also making sure that all of the outcomes for this project, as they start to come through, and we are getting more and more papers, which is fantastic, but making sure that we're able to share those both with the participants of the original studies, but also with the members of the public who are part of this project. Now, that's enough for me, since it's always quite boring to hear from PPI leads. I think it's much more powerful to hear from the members of the public who are actually involved with us. So it is my very great pleasure to hand over to Chris, who's going to share with you all a bit about his experience of being involved. So Chris, over to you.
Thanks very much, Amanda, and to Sarah, of course, and to all of you for these few moments, what will be a whistle-stop tour, I think, of my experience in PPIE. Public and patient involvement and engagement. So that started from me being a blood donor, and I was invited by Sarah Fowler onto her blood and transport research unit PPI group at the tail end of 2019. Now, in 2020, as we're all aware, quite a lot happened, and I think that rather accelerated the pace of PPIE work. So I got more involved. And right at the end of that year, the beginning of this year, quite unexpectedly for me, I was asked by Sarah if I might just be interested in attending possibly just one meeting of the multi-omics steering group. I said yes. I'm not going to attempt to define, particularly in front of this audience, what omics is all about. But what I will say is that the steering group is a group, well, in my terms, they're a group of very senior scientists coordinating the national reach of multi-omics activities from Cambridge University, the Wellcome Sanger Institute nearby, and elsewhere. So that's what I sort of got myself into. Thanks, Sarah. In terms of the role, I just want to begin by saying that for the multi-omics steering group, there was no tightly defined job description. I am the only volunteer lay representative, and so far there haven't been that many meetings that I've been involved in. I started, as I say, in January. So what I try to do in my role is what I would do anywhere in PPIE activities, to act as a public independent voice in general. And that's, I have to say, mostly based on my own interests, perceptions, and experience. So that would cover things like data protection, transparency, and accessibility of data, and a particular interest of mine because of my professional background in child rights development. So Amanda was talking about, becoming more diverse, seeing it how we can get more younger voices involved in PPI work. As far as the steering group is concerned, my role is mostly naturally based on the group's activities. I try to contribute to them as best as I can, for example, by taking part in the meetings simply and giving my lay opinions on, kind of what's being talked about. One particular aspect is that Sarah very kindly gives me a lot of her time after the meetings for debriefing sessions. And what we try to do in those is, well, widen my learning, things that we've talked about, heard about in the group meetings, but more particularly to find out ways of widening learning more generally for PPIE. So that can include, for example, helping to find how to include more PPI, more relevant PPI work in some of the multi-omics funding and research proposals. It can include helping to develop some of the lay summaries that we've talked about a little bit during today. And of course, participating and contributing to public events like this. So what I've found I can bring to the role, certainly my own relevant professional experience and the public interface of omics. I would point particularly in my case to work in international development and child rights. Again, we've heard very much today about the international scope of omics work. I also have a background in community health promotion and community health research, which has its place, and in information science. Well, in my day, it was called librarianship, so you can see how long ago that was. Like all of us, I have a natural curiosity. I have also, not like all of us, zero specialist scientific knowledge, so nowhere to hide my ignorance when I'm in the groups. But sometimes that can be a plus, I've found. It gives me freedom to ask perhaps unexpected, perhaps even unexpectedly daft questions in the meetings. And also, going back to the sort of the wider learning idea, it's an opportunity for me to learn and hopefully to share some of what I learn about omics, which is a privilege and very enjoyable. I have to say that there are many things I can't do. So I am, as I said, I'm the only lay volunteer among mainly senior scientists, and I am unrepresentative of the wider public in totality. So, for example, I can't bring a patient perspective, and in census terms, I suppose I'd be described as the older middle-class white British male. I just want to conclude in a way with what I think is the most important thing, that what I have found is the strong willingness of the senior scientists I'm working with to increase PPIE engagement and the public understanding of their works. And, in a fundamental and equally highly specialized research area with such profound links to human health as omics is, I really think that is a true example of good practice. So just returning to where I came in as a blood donor, just like my first experience of blood donation, I found being involved in the omics steering committee welcoming, supportive. It's opened my horizons. And if there's one downside, it's just that in the exclusively Zoom meetings we've had so far, the quality of the refreshments has just not been up to what I've expected at the West End Donor Centre. Thanks very much.
Fantastic. Thanks very much, Chris, Amanda, Sarah, and we are incredibly grateful to all of our public and patient representatives who've helped in many ways, as Amanda laid out, in terms of helping us with grant applications, with summaries of papers, with contributing to the design of projects and the ongoing running of projects, and particularly Chris, who, as you say, was thrown in at the deep end and has been our sole representative so far on the steering committee. And I think that talk really illustrated the challenges we have. I think at the start of this, we maybe hadn't really thought a lot as scientists about how we were best going to engage and involve the public in multi-omics research. So we're on something of a learning journey, and we've been ably guided by Amanda and Sarah, but I think it's something that we're still figuring out how we can do best, and we're very keen to improve on that and to hear the experiences of our patient and public representatives to help guide us in that. So we look forward to that. Certainly an area I'm sure we can improve on, and we look forward to working with our panels to do that in future. Are there any questions? Let me just have a look in the chats that people have. I see we have a question from Diana about, from the patient perspective, about kind of questions of agency and control, about the sort of sensitive information that's there, and whether there's any concerns about loss of control of health data and electronic health records. I don't know if that's something, Amanda, that's already been sort of touched on in training, or would be a good idea for a future workshop, perhaps.
I think so. I think most of the, well, all of the data that we're using within this project is de-identified. So I think the overall risk, as I understand it, is pretty small, but I think it would be useful for Sarah and I to return to the various different ways that data are safeguarded for this, because there's multiple layers of safety, and I think it's important that we are reassuring where we can be.
Absolutely, and I think as Chris mentioned, the world has changed a lot in the last year with the pandemic, etc., and certainly with data linkages and data usages. And I think being always ensuring that we have the safe use of data at the forefront of our minds is important, and particularly as data access is changing and the uses of data are importantly changing too. Excellent, thank you very much. We're going to move on now to Albert Tenesa from the University of Edinburgh. Albert's a professor of quantitative genetics in Edinburgh, part of HDR UK Scotland. So Albert, over to you, please.
Okay, let's see how it works. Can you see the right screen?
Yeah, that's great. Thanks, Albert.
All right, okay, thank you very much. So, good afternoon. So today I'm going to present some of the work that we've done for the last 2 years or so. And I will start to answer the question that I was asked to have. So why is this research important to the public? And I think the reason is that we really want to understand the interpersonal dynamics on health risk behavior. We need to know how to measure it, how important it is, probably more importantly, whether we can modify it and how to modify it. And also then, if we can modify it, how to assess the cost benefit of that change. And this talk is really, in truth, about the environment. And the environment is not really difficult to measure, because a glass of wine, that does not mean the same to everybody. Okay, so we've been measuring the same environmental risk factors for decades, and we know a lot about them. But the problem is that we need to know what to measure, and that creates some limitations on how we deal with the environment. In complex traits or complex diseases, so these are traits or diseases that are determined by a large number of genes and a large number of environmental factors, usually of very small effects. So we need large sample sizes to detect those effects, and that's why we need to accumulate data from multiple cohorts, even from even multiple parts of the world. And the model that we work on is that there is a direct effect of our genotype here expressed in different colors for each person, plus our own set of environmental risk factors. And this determines how we behave, the diseases that we have. And we call that direct effect of our genotype on ourselves the direct genetic effect. And that's been what everybody has studied for years. However, I think things are slightly more complicated than this. So other people, our siblings, our colleagues, our parents are actually a very important part of our environment. Humans, as we know, are very much a gregarious species, and that means that social interactions are actually very important. So in this plot, we can see, for instance, that the two ladies in the middle, one is part of the environment of the other, and that the lady to the left has as part of the environment the other two people to the right side. Okay, so but then one can think, well, if you just told us that measuring a glass of wine is difficult, or measuring it in a consistent way, how do we measure the people as an environment? And here is where the genetics come in place. So we can use the genetics of each person, the genome of each person, as a proxy of the environment that person exerts on other people. And what are the advantages of measuring the part of the environment as the genotype of other people? Well, there are, I guess, three main advantages. One is that we can measure it accurately, consistently, so we can measure everybody in the same way. And as time has gone by, and probably it's going to happen even more in the future, it's becoming increasingly cheap to measure genotypes. In addition, genotypes are constant over time. We need to measure them only once, and actually they can be measured from birth, or even actually earlier, but before birth, but probably less complicated to measure from birth. And we call the effect of those genotypes on someone else indirect genetic effects. So we have a combination of direct genetic effects and indirect genetic effects. And in previous work, most notably by Kohn et al., they looked at indirect genetic effects by testing the association of not transmitted genotypes from parents to children. And this actually is a very, very elegant work and clearly show that there are cross-generational effects of the genotypes that are not transmitted to the children. However, there is also the possibility that these effects happen within generation, between, for instance, members of a couple, or between siblings. We all know of siblings rivalry or cooperation. Those kind of effects have a function on the phenotypes of the siblings. So we started by we wanted to know whether there was evidence of horizontal indirect genetic effects, and we wanted to know whether they are common or they are just restricted to a few traits like education attainment, or even what we call maternal effects on growth. So the growth of a baby on the mother's womb depends in part on the genes on the mother. So we wanted to study these two questions, and to do that we actually selected 105 traits. We tried to select a wide range of traits. Some of these traits are expressed on two members of a couple. Some of them are only expressed on one member of the couple because they are sex specific, like prostate cancer or breast cancer. And we selected a few which are shared between the couple. So for instance, the number of children or the number of vehicles in the household as a way of testing what would happen for those kind of traits that are shared between two members of a couple. So this is the distribution of the traits we selected. So mainly are disease traits, psychological traits, and diet and lifestyle traits. And what we did was we used a very a technique that is used to estimate something that we call heritability, which basically quantifies how important are genes in the variation that we see in disease risk or on a trait of interest. And we model those traits as a function not just of the person genotype, but of the genotype of the person that is cohabiting with them. And we could call that the direct effect, here shown in big circles as the direct the own heritability, and the direct effect as the partner heritability. And we estimated that for 105 traits in UK Biobank. So we found that for about half of the traits we found a significant effect of the partner's genotype, and about a third of them had statistical evidence that accounted for the large number of statistical tests that we had performed. So that was good news. So we shown that the genotype of a person affects not just their own genotype, but that of their partner. And then we had to do a number of housekeeping and checking that everything worked okay. So the first thing that we did was to check that genotype of the partner was actually eating part of the pie of the environment. So if you can see this pie, where you want to show that it's removing part of the environment, not part of the genotype of the variation that is determined by the genotype. And then we did a number of things to just reassure ourselves that the results make sense. So we wanted to check that the data couldn't be explained just by another phenomenon called assortative mating, which means that people have a predisposition to partner with people that are similar to them in phenotype. And there are a number of other things that we wanted to check, to whether permitting the partners would lead to the same effects, and whether we could actually improve prediction of the phenotypes in siblings and mixed couples, in addition to using their own genotype. I will only talk about the first point briefly because we really don't have time. So the reason why we believe that this cannot be explained only by assortative mating is that if there is assortative mating, we know that the genetic theory predicts that the correlation, the square correlation of the square of the phenotypic correlation times the heritability of the person's own genotype should follow this line. Okay, so if the data could be explained by assortative mating, all the points that I will show you next, which should fall on this line. And that actually doesn't happen. So as you can see, all the points are above those lines, and that suggests that those effects exist. So to conclude, about half of the traits had significant partner heritability. About a quarter of those are likely to be influenced by indirect genetic effects on top of associative mating. I think genotypes are a good way of measuring environmental exposure, but we need better study designs and large sample sizes to better separate effects of indirect genetic effects and associative mating. So in terms of what is next, so I would like to try these things with simpler intermediate traits like the omics traits that the other speakers have talked about, because they are closer to cell and tissue function. I would actually like to understand to what degree, what is the relative importance of vertical and horizontal indirect genetic effects. So for instance, are they causal? Do you know how can we prove causality? And also things like that we don't know, like are indirect genetic effects from parents wear off with time, and how quickly do they wear off? And that's it. So I would like to acknowledge my co-workers on this, Charlie Shea, who did most of the work, and Oriol Canela and Conrad Rowley, and obviously our funders. So thank you very much. I'll answer any question that you may have.
Thanks very much, Albert. It's an interesting topic that hasn't been, I think, widely considered, and certainly not within the multi-omics community. And I think the idea that you present at the end there of seeing whether or not these indirect genetic effects might also have an impact on the kind of multi-omic traits that we've been talking about today would be a valuable question, and understanding how that differs perhaps from some of the other phenotypes or diseases looked at in UK Biobank. In terms of, I don't think I see any specific questions here in the chat, but in terms of looking at this in a multi-cohort way, do you see any challenges to doing that across a number of cohorts rather than just within, say, one cohort like UK Biobank?
Yeah, so we would have the same kind of issues that we've discussed. So how to homogenize the data, how to account for structure that there might be between cohorts. So I think some of the cohorts that have this kind of data are actually much better than UK Biobank, although smaller, because they have a lot more of family structure. So it is better, they are better suited to separate sibling effects and couple effects, for instance. And the question is how we could run the analysis on all the data together, assuming that we would have a unified phenotype, and we were working on methods on how to do that. So we've been working with distributed, with a way of distributing data across different servers, and whether that can be shared in an anonymous way.
I see.
So we're working on that.
Okay, well, I think that would be an interesting area to explore in the multi-omics cohorts community, and look forward to discussing further how we might be able to do that.
Yeah, that would be great. Thank you.
So for the last portion of the day, we thought it would be remiss of us not to, as we sit here mostly at home during a global pandemic, not to mention some of the work that's gone on in COVID-19, which of course a year ago we were only just starting to think about how, or starting to kick off this project. And when we wrote the grant application for this, we were talking about diseases like heart disease and diabetes and fatty liver disease, other diseases you've heard about today. And we hadn't anticipated how we might transform our data and methods for COVID-19. But I think in the last two talks, you'll hear some excellent examples of ways in which we've quickly changed direction to do that, and in a relatively short space of time, produce some really interesting science that's aiming to tackle some of the questions around COVID-19. So the next speaker is Liam Gaziano, who's a PhD student both here in Cambridge, but also in Boston in the United States, from where he's speaking today. So over to you, Liam.
Great. I'll share my screen. So as Adam said, I'm a PhD student working with the Cambridge Cardiovascular Epidemiology Unit, but I also work with some collaborators in the Million Veteran Program here in Boston, and this is one project that's kind of resulted from that collaboration between the Cambridge CEU and MVP. Of course, I'm presenting a paper that we recently was published in Nature Medicine, and of course there are many people involved who played a part in this role, and I'm just one of them. And so here are the list of people, many of which are probably on this call now. So what we did is here's kind of the overall analysis plan, and what we did is we started with trying to identify all proteins that are immediately actionable, meaning that they have a protein, they have a target for that protein that can be administered immediately. We felt that the need for therapeutics to treat COVID was pretty urgent, and so we said, let's refine our question to just looking at actionable proteins. And I'll go over this briefly and then go into all of this in more detail. Then we built genetic instruments that we can use for Mendelian randomization in kind of publicly available data, and then ran the MR, and then ran a bunch of follow-up analyses, which I'll get into later, and particularly co-localization, and I'll show kind of the importance, I think, of co-localization after MR like this. So this is some collaborators from the Kemble group, specifically Anna Galton and Andrew Leach, helped put together this list of limiting the scope of the question from 20,000 kind of protein coding genes down to just 1,263 that were kind of targets of drugs that are currently approved or in are clinical candidates. That again, that was to allow these results to be translated relatively quickly. We don't have 10 years for a drug development program for COVID, and especially at the time we started this analysis before we had vaccinations, and then still that's probably a good chance for COVID is here to stay, and we need drugs to address it long term. So we refined the question down to 1,263 unique proteins, and this is kind of a good illustration of the MR using kind of EQTL instruments from GTEx. This is from a paper in Nature Communications by the group in Bristol, which I think has done a great job here of illustrating how a good approach using kind of Mendelian randomization using EQTLs. So what they did is, and what we did is select instruments in one gene in one tissue, and then test that against your trait. In our case, it was COVID-19 hospitalization, and then iterate that for each gene in each tissue. And present them all. Some things we did slightly different is we used kind of multiple EQTLs rather than just kind of the peak EQTL, but still a very similar approach. And so these are the results when you run that MR using kind of the GTEx EQTL instruments. we then took all of these results that kind of passed this blue line threshold and performed co-localization. And from that, 3 genes really popped out: ACE2, IFNAR2, and IL-10RB. So let's go into some of those. First, I thought it'd be worth kind of, I think, illustrating the importance of co-localization, and I'm sure I'm not convincing anyone, or a lot of people on this call don't need any convincing. But here's an example of CCR1. So CCR1, if you use the EQTL instruments and test it against COVID-19 in MR, that produces a pretty strong MR result, and you might be convinced that CCR1 is involved in the disease, which it might be, but there's also, I think, evidence to show that it might not be, particularly if you look at kind of the co-localization between these 2 plots. So on the top right here is the regional association plot. So for those maybe not as familiar, that what we do is we plot the position of the variance on the x-axis, and on the y-axis the negative log of the p-value. And you can see that the peak variant here for CCR1 is certainly not the peak variant down here in the bottom for COVID-19. What you'd like to see is which is the other kind of co-localized results show, is that the peak variant in the expression data is at least in somewhat LD with the peak variant of the outcome data, and they kind of nicely line up. Here, obviously the peak variant for COVID-19 is way off in this upper left corner. So, some indication that maybe this MR result is actually by, due to kind of this LD confounding by LD, and not necessarily kind of a shared causal variant between the expression data and the outcome data. So sorry for the technical bits. I'll try to show more of the clinical implications of these results, but here is one example where they do kind of co-localize. So here you can see that the peak variant, the peak EQTL in the top right kind of is also the peak variant in the outcome data. And this is obviously big biologically plausible result, and we kind of thought it as a nice kind of positive control. Not terribly surprising that ACE2 would pop up in these results. It's the protein that binds kind of the spike protein from the SARS-CoV-2 virus, and there is some experimental data, some trial data on whether this kind of recombinant ACE2, one mechanism people suggested is kind of flooding your system with recombinant ACE2, which can maybe mop up some of the spike proteins from the SARS-CoV-2 virus. There are some initial reports, this is in Lancet, Respiratory Medicine, that showed kind of reduction in viral load. Perion, the company that kind of put this trial together, recently put out a press release that said that their kind of phase 2 trial was somewhat successful, was relatively successful in reducing viral load. So that was kind of a nice and maybe positive control, but we spent a lot more time kind of looking at this region called IFNAR2 and IL-10RB. The one problem there is that the EQTLs for both, these are 2 genes you can see in this figure that lie next to each other, and they're EQTLs, so variants that affect the expression of both IFNAR2 and IL-10RB, were also highly associated with COVID-19 hospitalization and co-localized. And this is not a terribly uncommon occurrence in kind of expression data. In regions you often see this kind of variants that are associated with multiple, soluble phenotypes or, genes in that region. And it can make it difficult to interpret, well, which one is actually driving this association with COVID-19 in the MR. And so this is why we spent a lot of time kind of doing these follow-up analyses to see whether we could disentangle. And sometimes I think you can't. I think we tried here and I think fairly successfully, but sometimes I think it's impossible to disentangle kind of this horizontally pleiotropy results. But here we'll show you kind of where our reasoning came from, where we ultimately thought that IFNAR2 is actually kind of driving the association with COVID-19 hospitalization. So the first thing we did was looking at GWAS, so phenome-wide scans of kind of these variants, these EQTLs that are EQTLs for IFNAR2 and IL-10RB. And we found that these variants, affect expression of both, but also affect protein levels of these two proteins that are known to be affected by kind of administration of type 1 interferon. So the treatment for the drug target for IFNAR2 and IL-10RB are type 1 versus type 3 interferons. So these are recombinant proteins that attach the receptors. And so what we found is that a lot of these variants mimic, or this variant kind of mimics antagonistic effects of type 1 interferon, which would kind of lead to us to think that IFNAR2, because that's the receptor for type 1 interferon. So next we put we did kind of this pathway enrichment analysis. And I think the key takeaways here is, again, it's pointing towards kind of type 1 signaling. There's a lot of nice results that pick up kind of viral defense mechanisms, but also mostly a lot of type 1 interferon, like gene ontology response to type 1 interferon, and not so much type 3 interferon. So again, adding a layer of evidence that maybe we're mimicking the effects of type 1 interferon signaling and not type 3, which would lead us to think that IFNAR2 is the more driving force, the protein with the driving force for COVID-19 hospitalization. And then the last piece of evidence is that IL-10RB is measured on one of the O-link panels, which was measured in about 5,000 participants from the O-link, from the interval study here in Cambridge, or there in Cambridge. And we found that those PQTLs from kind of plasma that measured plasma levels of IL-10RB were not associated with COVID-19. I tend not to think that a negative MR is necessarily meaning that the trials will be negative, but I think in this case it's just another layer of evidence that maybe IFNAR2 is the more likely candidate here. So
1 more minute, Liam.
Yeah, only 2 more slides. So then lastly, there are some human data to support this. So here's a paper in Science that showed that antibodies for type 1 interferons were found in a much higher proportion of individuals with severe COVID-19. So, building a case, but then there was a bunch of trial data that came out and said, well, actually, this kind of type 1 interferon is possibly, harmful in kind of later stages. And I think that fits in nicely with the data, or the data kind of seems to suggest that this should be administered kind of in the early phase of viral response. There seems to be pretty distinct phases of disease for COVID-19, and you can see why things can have opposite effects at different stages. So that's the last slide.
Fantastic. Thanks very much, Liam. And it's a nice example of how several different UK and US and in fact international cohorts can combine data on COVID-19, on gene expression, on protein levels, etc., to try and identify potential causal pathways and indeed drugs that could be helpful. As you say, perhaps with challenges around at what point in the disease time course the drugs should be used. I think in the interest of time, we'll move on to the final talk, which is from Lucia Claridge, who's a UKRI research fellow at the University of Edinburgh, who's been working with similar data and cohorts to answer a similar kind of question.
Hi, thanks, Adam. Just a second. This part can always be a bit stressful. So do you see my screen now?
We do. We see the, yeah, there's the.
And it's still working. Perfect. Okay.
Yeah, that's great.
Very much. I would like to spend last 15 minutes of this meeting to show another example how integrating together different omics datasets, in this case genomics, proteomics, and transcriptomics, can help us understand mechanisms behind complex diseases. So you might think now with vaccination there's very little need for further treatments for COVID, but we know still that unfortunately some people still end up in hospital and still end up unfortunately dying. So we still need to find new treatments for this disease. And to do that, we would really need to understand what's the underlying biology that happens behind this disease. So we need to understand what mechanisms are happening in order to suggest and provide better treatment for these patients. And I particularly like proteins to help to understand the underlying biological mechanisms, because proteins are main functional units. Anything that happens in body, there are proteins involved there. And so when we measure these proteins, plasma proteomics in blood, we are basically getting a snapshot of what's happening in our body in various different aspects, in different tissues and different cells. So they reflect diverse biological processes. And they are particularly nice because they are really good targets, and already for many of the proteins that we measure, already some targets exist. So we also have a good potential of repurposing existing drug for a new disease. So overall, studying changes of plasma proteins in COVID-19 might help us understanding better the biology behind it and help with the treatment of this disease. So to do this, we first started by asking a question, well, are there any proteins that are associated with COVID-19? And for this, we took two studies that used all-link proteomics. One used 306 cases and 78 controls. The other one had somewhat smaller number of cases versus controls. But in common between these two studies is that they both came out with a list of proteins that they consider are associated with disease. What we did then, we took the overlap of these proteins, and we found that 157 proteins, when looking at these two studies together, are suggested to be associated with COVID-19. But now the question is, are these proteins causing the COVID-19, a severe form of COVID-19, or is COVID-19 changing these proteins? So which way around this happens? Because and in order to disentangle this question, we use a method called Mendelian randomization analysis, which you've heard of a few times already today. This is a very neat statistical trick that uses the fact that genetic variants that we have are randomly assigned to us at conception. So we get half of our DNA from our mom and half of our DNA from our dad, but which part we get from whom is sort of random. And this is very good to help with reducing the confounding. What do I mean by that? Let's think about BMI for a minute. So if it's people with higher BMI tend to also have higher likelihood of having hypertension. So does that mean that BMI causes hypertension? Well, not necessarily so, because as you know, people with higher BMI might also have different dietary preferences. They might eat more sugar or more fat. They might exercise less, and so on and so forth. So all these other things might actually be confounding our association with BMI and might actually be causing hypertension rather than BMI. However, if we look at the genetic predisposition for higher BMI, this shouldn't be confounded by these all external factors, because as I said at the beginning, the genetic variants that we have, we get randomly from our parents, and what we eat does not influence our DNA. So if we have higher predisposition for having high BMI according to our genetic variants, and we still have higher likelihood of getting hypertension. That might implicate that BMI is actually causal for hypertension. So we use this neat trick on our COVID project, where we actually tested, does genetic propensity for higher protein levels of this 157 that I already mentioned, results in higher risk for severe COVID-19, our outcome. And if that's so, there's a higher likelihood that that protein, specific protein, is causally involved in the disease. To do this, we need to have, or also already mentioned, genome-wide association studies of each of both exposure and outcome. And here we were really lucky that we are part of the Scallop Consortium. That's an international consortium of 42 cohorts across the world that has all in proteomics. And through this consortium, we got early access to genome-wide association studies of 157 proteins in a big number of samples, from 2,500 to 26,500 individuals. On the other hand, we took severe COVID-19 genome-wide association studies provided from Host Genetics Initiative, and they were particularly interested in looking at the analysis where they looked what are the genetic predisposition for requiring respiratory support in hospital or dying due to disease. So what we call, what I'll call from now on, severe disease. So using these two data sets, 157 proteins versus severe COVID-19, we ran Mendelian randomization analysis, and we come up with 5 proteins that have significant MR causal effect estimate for the severe COVID-19. But however, as every statistical test, there are some assumptions behind Mendelian randomizations, and I won't go into the details, but we performed various tests to check if we are satisfying these assumptions, and this resulted in 3 out of 5 proteins falling out, so meaning that assumptions were not satisfied, so we can't really trust this estimate of causality. But the 2 proteins were still significantly significant. So then the next question you ask when 2 traits are associated with the same genomic locus, what was also already mentioned in the previous talk, is colocalization, meaning if I have protein and that's associated with specific genomic region, and I have COVID-19 that's associated with specific region, do their local associations overlap, meaning that there's the same underlying causal variant causing both, or they do not overlap, meaning that there's a different causal variant affecting one or the other. And why would that be interesting? Well, if you have the first case, having a drug target that influences protein levels might help reducing number of deaths from severe COVID-19, while in the other case, reducing the protein levels it's not likely to have much of an effect. So with the remaining 2 proteins, and this is called colocalization, and for the remaining 2 proteins, we next ask, so do they colocalize with severe COVID-19? And here on the left-hand side, you see that FAS levels of the protein FAS neatly, nicely overlap with the levels of COVID-19. On the other hand, for the other protein, we see on the top panel that protein peak is completely separated from the COVID-19 peak, meaning that this protein is not likely to be causally involved in COVID-19. So this FAS protein is quite interesting protein because it's part of the tumor necrosis factor receptor superfamily. It's interesting because it has important role in programmed cell death. That means our cells with time just get old and they need to be killed, or something goes wrong in a cell and this cell needs to be removed. This process is called apoptosis. And so this protein, that's membrane receptor, is involved in apoptosis of particular lymphocytes, which are blood cells involved in immune response. Interestingly, if you remove this protein from a mouse, they develop autoimmune disease that's similar to lupus, human lupus. And if you have malfunctioning form of the protein, if you have deleterious mutation in the FAS gene, people tend to it results in a rare Mendelian disease called autoimmune lymphoproliferative syndrome, meaning that there's too many lymphocytes, there's no limit on how many lymphocytes there are. So there's defective lymphocyte apoptosis, or there's too many lymphocytes, which results in the autoimmune disease. So next question that we ask is this protein associated with just severe COVID or COVID in general? And so we repeated this analysis with 2 different type outcomes of COVID. One is hospitalization. So those were all the patients that were hospitalized due to symptoms associated with confirmed infection, or susceptibility. That just means whoever had confirmed infection with or without any symptoms. And you can see here that it does really seem to be associated with severity itself, because we see the effect is very small in susceptibility, a bit higher for hospitalized cases, but still not significant, while it's really significant in the very severe form. So next question that we ask, what's the proposed mechanism behind of this? So is it just the expression, change in the expression of gene levels of FAST results in a change of protein levels? And for this, we looked in the gene expression data from 2 public repositories and various tissues. And here you can see a similar plot. Now on the top, we have a protein levels, while on below we have gene expression in whole blood and gene expression in lung. And by now you can maybe already start seeing that these are not really matching perfectly. So it seems that the gene expression has different causal variant compared to underlying causal variant compared to the protein. So genetically increased levels of FAST proteins are not increasing the risk for severe COVID-19 through gene expression. So that was a bit of maybe a surprise. So, but then we went and looked into the protein itself. This protein has 4 domains: extracellular, so that's the domain that's outside of the cell and that receives a signal if a cell has to be killed. It has transmembrane domain. This is the domain that keeps it fixed at the membrane. Intracellular domain that contains the death domain. That's the part within the cell that when the signal on the extracellular comes, signals the cell that the cell needs to die. This is encoded by a gene that has 9 different exons, and when you have all 9 of them, you get this top picture with full protein that's membrane bound. However, when this 6th exon is missing, this transmembrane domain is missing, which means that this protein doesn't have this anchor for the membrane, and it's soluble receptor, so it's released in the bloodstream, it's secreted. So the next question we ask, is the change in this specific transcript, so this protein that doesn't have exon 6, is that results in the change of the protein levels that we are detecting? And here our friends from the Interrogating came to help using their RNA-seq data in 4.7 thousand participants. We checked for associations with level of this specific version that doesn't have the exon 6, doesn't have a transmembrane domain. And you can see here now, again, we can have a protein top panel and skipped exon 6 transcript on the lower panel. And you can see here that these 2 match, suggesting that genetically increased levels of soluble FAST protein, transcript site and exon 6, are increasing the risk for severe COVID-19. What does that actually mean? Our proposed mechanism is so that individuals with a C allele in this particular SNP, they have more of the full transcript, so they have more of the FAST membrane-bound FAST protein. So when the FASL, this is FAST ligand, it's a cytokine that brings the signal of the cell having to die. So when FASL arrives to the cell membrane, the apoptosis program cell death is started. However, individuals with the T version of this allele have more of this shorter transcript that lacks the transmembrane domain, membrane anchor. So what happens that these soluble FAST proteins steal away, they serve as a decoy receptor for the FAST ligand, so which results in the reduced apoptosis of, we think, probably lymphocytes. And so these lymphocytes, there's simply too many of them, so they're increasing the inflammation, and then through the increased inflammation are increasing the risk of severe COVID-19. So all in all, I hope we show that understanding molecular pathways can help with treating the disease, and proteins might be really good proxy for that, because they provide a snapshot of various molecular pathways and processes that are happening and are also at the same time targetable. Sorry about this. So by performing Mendelian randomization analysis, we show that propensity to increase levels of soluble FAST influences COVID-19 severity. And we propose the mechanism that this happens by just simply this soluble FAST protein stealing away the decoy cytokine, reducing apoptosis of the lymphocytes or maybe virus-infected cells, which leads to the increased inflammation and risk of severe COVID-19. I didn't mention, but there's also potential for drug repurposing here. There's an existing drug called ibrutinib, which is tyrosine kinase. It's an inhibitor that's used in treatment of hematological malignancies, and it has been shown to decrease soluble FAST, so thereby enhancing FAST-mediated apoptosis. So it no longer happens that, so basically it steals away the soluble FAST and then enabling back the apoptosis. But with apoptosis, we have to be really careful since it's very important process in many other cells. So let's see, it still needs to be remained checked to see if it's really going to be a useful treatment or not. And I would like to still last minute, if I can, more for a bit of a shameless promotion. Our two cohorts from the north of Scotland on Adam's map from this morning, Orchids and Viking, are really well annotated cohorts for different omics datasets, and we would be really happy to contribute to more of the international and UK-wide efforts. As you see here, we have genomics that you heard a lot about, metabolomics we heard a lot about, proteomics, but we also have methylation and maybe uniquely glycomics. Not many people have glycomics, and we have this in from 1,000 to 2,000 people, and in some of them in both cohorts, which gives a really nice data set to test out different things. And all these people are also connected with their electronic health records. I would like to end with thanking people involved in this project. So the people in blue are the core analysis team. Jimmy from Imperial College of London and Jim from University of Edinburgh, and Nicola and Marisa from Edinburgh, Jack and Artemis. We were all involved for the last 4 months, day in, day out, working on this project to try to get these results out as soon as possible. Also, many thanks for people from Cambridge that helped and provided the data for this, that finally pinpointed the mechanism of this skipping of the exome, and then all the cohorts that contributed proteomic data, Scallop Consortium, but particularly Erin and Jinghua, who provided early access to their meta-analysis efforts. And that's all. I'm happy to take any questions.
Fantastic. Thanks, Lucia. Great way to finish. And I think really nice to highlight there that this has been a really intense project, but only over the last 4 months or so. And it's really perhaps exemplified what can happen when people across the country and even beyond can kind of pull together in the same direction for a very high urgency project. And these kind of projects that typically would take us much longer to do, but have been really rapidly advanced because of the urgency of the COVID-19 situation. And I think also a nice illustration in those last 2 talks of how we might use the kinds of data we're talking about here to provide potential ideas about which drugs could be used in trials, which of course has been a major issue over the last year of how to decide which drugs to test in trials related to COVID-19. And I think this is where some of the multi-omic data could be really useful, not only for COVID-19, but for other diseases. There are 1 or 2 specific questions in the chat, Lucia, that might be helpful to perhaps address in the chat, some technical questions around SNPs and co-localization. But I think as we're drawing to a close, it would be good just to thank everybody for attending, particularly thanks to all the speakers for their excellent presentations and showcasing some of the fantastic science that's going on around the country and beyond. We've seen science that's involved many of the different cohorts that I showed on the map at the start, both across the UK, but also from other parts of the world as well, China and the US in particular that we've heard. We've heard about many different types of multi-omics, not only proteomics and metabolomics and genomics, but also measured on different platforms as well. And one of the challenges that's been illustrated is how to integrate those and whether we can really consider measurements made on different platforms together. And I think we heard nicely about how that sometimes works very well and sometimes is more of a challenge. So I think some challenges there for us to continue to rise to. We've seen unpublished work. We've seen work that's been published very prominently in high impact journals, which is fantastic. And some of those talks that you've seen are work that's currently being considered at some of the top journals. So we hope to see those out in print in due course. I set out at the start of the day kind of 3 goals for the day. I think we've certainly celebrated today our successes. I've certainly also seen some challenges that I think we can try and rise to over the next year of the consortium, both in the scientific side and also in the engagement and involvement side. And certainly plenty of opportunities for enhanced collaboration, not only with additional cohorts, but with additional types of methods that kind of Albert talked about, but also existing projects which could clearly benefit from the inclusion of more data. And so thinking about how we can do that going forwards, and build better connections and bring together more of our data sets for these kind of questions will be very important. I also need to thank Amy Lafont and Sarah Foley, who've put together the whole day today, really, and done an enormous amount of work behind the scenes to make the event run smoothly, which it has. So thank you to both of you for all your hard work in making the event happen. Thanks to everyone as well who's been involved in the omics Cohorts Consortium so far for all the fantastic efforts, particularly the work package leaders who've helped drive the science forward. The fantastic coordination team here in Cambridge who help everything move behind the scenes, and also, of course, all the patient and public representatives who have helped us drive the project, to shape the project, to help enhance our communication and engagement, and certainly will continue to do so. Before we draw to a close, I wonder perhaps if John Dinesh would like to say anything on behalf of the Understanding Causes of Disease theme, which this project fits into.
Thank you, Adam. Very little left to say. Just to say what a superb way to spend 3 hours. It was inspiring. It's clearly an area of endeavor that this grouping, the UK, Health Data Research UK's Understanding Causes of Disease, is world leading in. I think what we heard today shows the power of the approaches, but also the versatility of the approaches, being able to tackle a range of questions around non-communicable and communicable diseases. I was really struck by how the multiple layers of the effort are expanding, not only just the genomic layer to a particular molecular layer, but as Adam mentioned, moving across platforms, across phenotypes, across cohorts, and across continents, as the efforts are now truly globalizing, as we heard from some of the talks. So what a tremendously inspiring session. Thank you to all the speakers, and special thanks to Adam and the central team for pulling it all together and orchestrating it so well. So I really enjoyed that, and I think it's a sign of great things to come.
Thanks very much, John. So I think the final thank you really is just to say thank you to all the participants for joining us, for engaging, for taking the time out of your day to sit and listen and asking questions. And the final couple of things to mention, again, just a reminder of the survey that we're hoping that we can get lots of answers to from the multi-omics community to help us design what are the ideal informatics infrastructures for multi-omics. The link is there, and we'll put the link in the chat again, and we'll be emailing and putting this out in various media channels. And finally, just some dates for your diary. There are several events that have appeared in the last few days on the HDR UK Slack channel. If you like your multi-omics, there's a workshop on molecules to e-health records that's happening at the start of May, and there's a Zoom link there. The HDR UK Annual Scientific Conference is always a fantastic event. That's happening this year on the 23rd of June. More information is on the HDR UK web page. And more imminently than that is the opportunity to nominate Team of the Year, Impact of the Year, and Early Career Lightning Talk prizes. And the deadline for that is next week. So do have a look. We've heard today some fantastic teams and impacts and some great talks. So I think there's plenty of scope within the multi-omics community to nominate there. And finally, moving beyond the multi-omics, there are other insight sharing days from other aspects of HDR UK, including, for example, the Better Care Insight Sharing Day, which happens on the 22nd of April as well. So lots more opportunities to hear about more great science from across the HDR UK community. So thanks very much. I'll leave this slide up in case people want to take down the details, but thank you all for joining, and look forward to engaging and collaborating more with all of you in future. Have a great day, everyone. Thank you. Bye-bye.